Hi, and welcome to Conversations with Scientists. I'm science journalist Vivian Marks. Today's episode is a sneak peek of the 2026 annual meeting of the International Society for Stem Cell Research, ISSCR. The meeting hasn't happened yet, and this is a chat about some of the things that will happen there. If you are a stem cell scientist, please stay tuned. If you are not in stem cell biology or you are not at all a scientist, please come along for the ride. This podcast is for anyone interested in science. The ISSCR annual meeting is in Montreal this year from July 8th to 11th. If you haven't signed up and you want to, well, do it. And yes, of course, you can still do that all last minute, the world being what it is and budgets being what they are. For now, no suitcase packing needed. We had a bundle of questions for the two co-chairs of the meeting, Dr. Fiona Doetsch from the University of Basel and Dr. Nozomu Yachie from the University of British Columbia, and he has a lab at the University of Osaka too. My co-moderator is Dr. Angela Parrish from Nature Communications, who has also been at Nature Cell Biology. Together, we ask about the meeting, some trends, some things patients might want to know, we ask about science more generally, and we do a word association game. Our guests talk about how malleable the adult brain is, also about new clinical developments. And you may have heard that the Japanese authorities have given conditional approval to two therapies based on induced pluripotent stem cells. One is for heart ailments, the other for Parkinson's. This is the first of this kind of therapy. Dr. Doetsch and Dr. Yachie talk about many themes, including, as Dr. Doetsch puts it, in vivo veritas. They talk about stem cell engineering, about careers in stem cell science, the role of computation and AI, and why biologists and mathematicians are welcome at this meeting. Manuscript editors from across the Nature journals were on the call too.
Hi there. Nice to meet everybody.
Thank you so much. People are still dialing in. We have, I always put out an invite to manuscript editors. Some people have children who are being unruly, but
Absolutely.
Children are fine. Pets are fine. Thank you so much for taking the time for this. I know it's probably annoying and takes you away from more important things.
Oh, it's great. It's actually a wonderful opportunity for us to share about the meeting and inform more people. So we really appreciate the opportunity too. There he is.
Yeah. Oh, look at that cool photo. Oh, we're going to talk about manga perhaps. Hi, Dr. Yachie. How are you? Thank you so much for taking the time for this.
Oh, of course.
I want to ask about those pictures on your wall, Dr. Doetsch. What is that? That is cool. One is...
Oh, yes. This is on the, let's see, the green one. That's the stem cell niche in the adult mouse brain. And those are actually the stem cells. And then on the one beside it is a photograph. These are pictures I took when I was a student, actually, of chains of newly generated neurons migrating through the brain.
Super cool.
Yeah.
I know it's a podcast. People won't be able to see it, but you've described it really well. So thank you. All right. Well, let's get started. This is the sneak peek of the annual meeting of the International Society for Stem Cell Research. And there's a co-moderator. Angela, you can introduce yourself.
Hi, I'm Angela Parrish. I'm a senior editor at Nature Communications, and I've also worked at Nature Cell Biology. And I look at manuscripts mostly on cell biology, metabolism, organelles, and certainly stem cells.
All right. And everyone else, please just keep in mind that when you speak, you should introduce yourself first, say something, whatever you like about your hobbies, but also about the types of manuscripts you enjoy and the ones that you take care of. And I'll introduce the people who are the major guests here, and we'll get started on this sneak peek of the meeting. So first, we have Dr. Fiona Doetsch, and I saw that you just got a very cool award. Well, it's in January, right? It's the Louis Jeantet Prize, I think.
Yes,
so you're at the
correct.
University of Basel. So is that all correct? Your affiliations and all of that?
Yes, I'm at the University of Basel at the Biotzentrum in Switzerland.
Biotzentrum in Switzerland. And sorry, this is the evening for you, so sorry for invading on that. Dr. Yachie, you have a list of prizes. Oh, I've got more people to admit. Excuse me. We've got a crowd. So, Dr. Yachie, I want to ask you about manga because your site has some very cool manga. But so you have a bundle of prizes I saw: Allen Distinguished Investigator Report, Nagase Prize, I think, of the Japan Society for the Promotion of Science. That sounds special. And others too. And you are, let's see, you commute between the University of British Columbia, Osaka, and Tokyo universities. I'm not sure how well.
Mostly between UBC and the University of Osaka. Yeah, like every month, like back and forth.
Wow, very cool. So
Yeah,
you have two lab groups, I think,
that's
right?
right. Yeah,
Very
that's
cool.
right. Yeah.
All right. So the reason, and I mean, it doesn't really feel like summer yet. I'm freezing. It's freezing in New York right now. But in July, there is this meeting, the annual meeting of the International Society for Stem Cell Research. It's in Montreal, Montreal, from July 8th to the 11th. And a few people are going to get together and talk about stem cells. And I wanted to ask, how many people are you expecting and how many talks? Numbers are always kind of fun.
So I think we're expecting around 3,500 people at the meeting. So it's a very large meeting, crossing all aspects of stem cell biology, from basic research to clinical translation.
And there's lots of posters, I think. I mean, I heard some numbers, but do you have an approximate count?
I think right now the latest numbers are around 1,400 submitted posters. And then the number of talks, I actually don't know the final number of talks, but from those abstracts, there are going to be 80 selected speakers, as well as the concurrent sessions and the plenary speakers who are invited as well.
Got it. And the concurrent sessions, just so people know if they can't make it for various reasons, are there ways to tune in? And for attendees, can they listen to recordings afterwards? I know hybrid meetings are a bit hard to organize, but I'm just curious.
So we don't have a hybrid meeting at the moment, but I believe that the concurrent sessions are recorded and provided on demand after the meeting. So you don't have to make the really challenging choices of which talks to go to at the moment. So you could just sit in one session and then catch up later.
Okay, interesting. And because you're in very different time zones, just a practical question. I mean, I have this a lot with interviewees. Dr. Doetsch, you're in Switzerland. Dr. Yachie, you're sometimes in Japan, but you're mainly in Vancouver. How did you work that out? You co-organized the program. I mean, I'm not sure what times you met at. I'm just really curious about formalities of that. Wow.
I exercise well and sleep well, and sometimes I don't fix my jet lag.
I see. So time is all
Right,
relative. Okay.
right.
Got it.
Yeah, I think we're very open about this. I think we realize that for putting together a meeting, you just work at fun hours and everybody gets to interact at different times.
Right.
Fabulous.
Yeah.
Well, tell us about, I mean, we can't go through the whole program unless you want to stay until very late, which is fine with me, but I'm sure it's not fine for you. But what are some of the things that you are really happy about? And oh, right, there was one thing I wanted to ask. So Stem Cell Technologies is a company and is a co-sponsor. I'm not sure if they were part of developing the program or how they were involved.
So they've not really been involved in developing the program. They are providing a large amount of support to the annual meeting. And we were recognizing actually Alan Eves and Connie Eves, who sadly passed away recently, as lifetime achievement contributors to the society for their enormous contributions.
And they are
But
with the companies, the stem cell technologies. Okay.
And Connie Eve was a professor at the University of British Columbia.
I see.
Mhm.
Sorry about their loss. So that I see. So you recognize them at the meeting. Cool and important, those things. So the meeting, it's a few people getting together to talk about stem cells. What are some things that you are particularly proud of the types of, whether it's keynotes or maybe it's a block of presentations? You can pick any topic that is dear to you or things that you are really happy about putting together for this meeting.
Yeah, as a scientist who has a computer science, computational biology background, I really appreciate that, these days, as in any field in biology, like stem cell field is also highly integrated with AI and computational biology. And I am personally super excited about covering those talks.
Or one of the things I'm most pleased about is that we have so many first-time speakers at the meeting. So we have about 80% first-time speakers at the annual meeting. So one of our goals was really to try and reach out to people and bring new perspectives to present at the meeting. And so I'm thrilled that we were very successful at this. It's a very international set of speakers. And from my sense, I mean, we think about stem cells in the adult brain. So I really love this concept of thinking about the whole organism physiology and how this impacts stem cells through life, interactions with the environment, and then how we can use these new technologies to explore these questions. So we really tried to capture everything from non-model organisms towards clinical levels and to bridge these themes.
One thing that has come up in previous discussions with for ISSCR and also for other meetings is this idea of, a big tent means that, for example, computational people and neuro people and cell biologists are at a meeting, but they don't perhaps organically move together except maybe in the coffee line. And I hope the coffee is really good at the meeting. But how do you kind of set up the opportunities for them to interact?
So I think we have themed concurrent sessions, but at the same time we design those sessions to have other elements in each talk. So we strategically selected speakers who are interdisciplinary in a sense. Yeah, so that's probably one way to explain the excitement of the conference.
I think also we've really tried to create more opportunities this year for people to interact. And so we have meetup hubs, we have science spotlight sessions, which are actually organized by the members themselves. And so these allow them to bring up emerging topics that are really fresh. And we are trying to have different kinds of panel discussions and even create spaces where people who might share an interest can come together and discuss these topics in a way. So really trying to provide the spaces for the larger community to interact and build bridges across the world, especially at this rather fractured time. It's very important to bring people together.
Angela, if you have any questions that you want to ask, because I have 292 or so left to go.
I think about the conference in general. I was interested in the choice to have 75% first-time speakers, looking at earlier researchers and, people in the beginning of their careers, and sort of, is this a reflection of changes you're seeing in the field? Is this a sort of more optimistic hope of what the field can become? Just sort of in general, what are your thoughts? Because it is very interesting to see that, most of the speakers generally are seasoned. So at a big conference like this, it is pretty cool to see that you've made it a point that's important. So if you could just expand a little more on that.
It's basically all of them, but in addition to that, it's probably a sort of mining of a superstar from the young generations. If you look at those big conferences, like we tend to pick like big shots to speak out and introduce their big science, but at the same time, we recognize there are lots of like emergent young scientists who are doing amazing stuff. So, we want to hear more from them.
I think also they tend to be more engaged with new approaches and new ideas than more seasoned investigators maybe. So it allows us to capture at the moment sort of emerging topics or sort of more technology-oriented approaches that can actually shed light on fundamental biological questions. So this I think provides a really fresh perspective.
Well, that's a bold choice and very encouraging too. I also saw that you have I think it's called GPS, Career GPS, Navigating Your Future session. Who is that for and how is that set up?
So that's actually been organized by the Early Career Committee. And so they have invited people who have had different career trajectories, who will share their experiences on the panel and then really have an open discussion with the members. And each year they organize one of these sessions on different topics, and they're always hugely successful and really provide a great space for people to discuss their concerns, their ideas, and opportunities.
I see. So they're kind of panels, but then there's a Q&A that isn't just, the last 2 minutes of the session will have some cues.
Is really a lot of question answer and time provided for that.
Nice. Great.
It would probably also be a really good networking opportunity for all of the people that are just, even in the audience with each other. It's
Exactly.
Such an important way to get your career moving forward.
And hopefully, I suppose, speaking for myself, is there coffee and cookies? But we'll see, right? If that's there too.
We're actually having a big all-attendee reception this year, which I'm excited about too. So hopefully there'll be lots of treats there as well.
Wow, treats are good. Oh, that's nice. All right, so more incentive for people to network and also to snack. So we'll take a little break and do the word game. We do this on these sneak peeks. It's a game where we present you with a word pair. Actually, in one case, we have a trio, but it's a word pair. You pick one, and then we move on, and we can return to some of those later. It's an homage to a French journalist called Bernard Pivot, who used this in his show in French television for many, many years. Stephen Colbert uses it, and it all kind of goes back to Marcel Proust, who used a questionnaire in one of his novels. So I don't have a drum roll, so I'll just do it with my hands for now. Angela, are you ready?
Yep, I'm ready.
Okay. Nozomu and Fiona, are you ready? So, for example, we'll ask cats or dogs, and we can start with that. Cats or dogs, you pick one, and then we move on to the next one. Also,
Okay, let's try.
it's fine. And if you don't want to answer, or if you think if there's something to say about it, or you don't want, you can just say pass. It's not a strict game show. It's a very fluid, kind of open show. So, all right. So, cats or dogs?
No right answer.
Dogs.
Depending on.
Cats.
Okay.
I'll just do the next one, and then we can switch off. Angela?
Sounds good.
Big conferences or small conferences?
Both for very different reasons. So I think, are we supposed to expand, or are we just, that's
It's
it?
fine. We can expand later. For now, I
Okay.
think it's just kind of like scrunching a lot into this one little session. So cool.
Wait, sorry. Are we supposed to pick one or can we say both?
As I said, the rules are pretty fluid, but if you want to
Okay.
pick both,
Yeah.
that's fine. So big conferences or small conferences?
Big
Poster or talk?
Poster for interactions.
talks.
Let's see. We had one. Oh, yes, this one. Organoids or gastroloids?
Those are the same thing.
Oh, no. Are they? Well, one, I guess, is more for organs, and the other is more for early development, or am I it could be totally fine that
They
I
are both modeling life systems using stem cells. So
okay.
Okay, here's one. This might be hard. Endoderm, mesoderm, or ectoderm?
Obviously, ectoderm.
That's what I thought.
all of them for me.
Yeah, without all of them, things don't really come together. That's true.
Ah, I see. Interesting. See, we weren't sure how you would react to this, but okay. What was the other one? Oh, yes. PhD, 4 years or as long as it takes.
4 years. Yeah, definitely not as long as it takes.
I don't know. I have mixed feeling about this.
That's fine. Mixed feelings are fine.
Let's see. Oh, okay, this one's interesting too. Stem cell or stem cell niche?
That's
came up
so
with these a
hard.
while ago.
Both.
Yeah.
They're so tied together.
Both.
Yeah.
But
Hard to have
probably
one without the
stem
other.
cell niche.
How about pluripotency or totipotency?
Totipotency.
Multipotency, yes.
Superb. We have more, but we can also call it the end of this game. But thank you very much for playing along. I appreciate that. And I maybe this gave people also time to ask questions. I don't see any raised hands as of yet, so we can keep chatting about those other many questions that we have. I was wondering about some of the answers, and I don't know if you want to talk about any of those. I guess the thing is really when you pick which model you're going to use, how do you decide, and how do you recommend others decide? Your trainees, but maybe colleagues, right? I mean, do you tell them this is the way to go, or do you say, no, I think your thoughts on this are welcome? Stem cell model, I mean.
I'm a type of researcher who wouldn't pick any specific model, but try to grasp the overall structure or concept of diverse models. And I started talking about this these days. So we are entering the AI era now, and we know the success of generative AI systems, which creates movies or which creates, photographs and so on and so forth. And stem cell is pretty much like that. It's a generative machine, and which generates diverse organs in the body. And there is a core principles encoded in the genome. So genome, in a sense, is a foundation model. So if you kind of started thinking about this kind of stuff, like everything is important, but more important thing is that if you look at different models, there are lots of commonalities, like common principles and rules, and we always chat, those commonalities, and this is especially important in the stem cell field. People are studying fly, people are studying human stem cells, but we often find common principles, and that's the excitement that we always have in this community. So,
The genome as foundational model. Oh, those are fighting words for some.
mm,
Okay, but cool.
So this is a kind of out there question, but then, so if that's true, and you can really find these underlying principles across these diverse models and put it all into its, whatever computer system, do you think
mhm.
you could then do all of the experiments you would need to do in the computer if you had all of the information and enough of this
Uh,
foundational information connecting them, right? These true
right.
principles from different models. Do
I
you think
think
you
yeah, I think that's quite possible, and that's what us, some of us are envisioning these days, including myself. The current generative AI is more like a black box. So we feed information and we generate the information, but we don't know the underlying principle. Biology has circuits encoded in the genome, and by feeding lots of data in biology to AI system, and somehow, we need a little more effort from computer science side. We may be able to find like the principles encoded, and therefore we may be able to simulate the human body in a computer. So,
I think that I love in vivo, in vivo veritas as a complementary perspective here. And I think it's fascinating how different people resonate at different scales. So some people are really interested in gene regulatory networks, some be really interested in cell-cell communication or the level of organs or the level of the whole body. And so for me, when I talk with people, I try and find what scale that they like to work at and think at and resonate with, and then come up with a question on those levels. And I think the exciting part now is really this idea of communication across not only organs, but within tissues, and how these intersect, and how you can have so many inputs coming together and being sensed by individual cells, and how they respond to these signals. So I feel that we have so many surprises still to discover, looking in these different organs in vivo as well. So the two approaches are very complementary, but we would always go and try and visualize and see what's going on.
When you made the finding that, I'm just going to simplify here because my brain can't do hard stuff, that neurons aren't just in the brain, aren't just they're there and they don't change, that there's actually a lot more plasticity in the brain than I guess a lot of people thought, maybe even colleagues of yours. How does that unfold, sort of from that first moment when you have this insight and observation and measurements, where you found something that really goes against perhaps some of the findings that many others have had, also the big shots that you've mentioned who are around, of course?
It's a really thrilling moment. I think there's nothing like looking down a microscope and seeing something that nobody has seen before. And then you just have to go with it and find more evidence to support your ideas, even if it's counterintuitive. And then you just design new experiments and you build up all of these lines of evidence. And then you keep looking for these big findings, or let's say just conceptual advances. And what does this mean? So, for example, we discovered a long time ago, actually, that the stem cells were a kind of glial cell, which was a big surprise because neurons and glia were thought to come from different lineages. And that was a tough initial path, but in the end, it's really it's transformed our understanding of how the brain can maintain plasticity through life. And I think another really key finding now is this idea that we have different sets of stem cells in the brain and perhaps in the body that respond to different signals. And at least in the brain now, if you can trigger these different stem cells, it provides a whole new kind of plasticity. So different physiological states can switch on specific sets of stem cells to generate rare types of neurons that may be important, for example, for in pregnancy, for a mother to recognize her own offspring. So what are the different states that can do this? So things are a lot more specified than we used to think, but also more plastic.
It's fascinating and interesting, the part about pregnancy that I know you've worked on. So I know that this is an exciting time in stem cell science because of some of the approvals of the Japanese authorities for certain types of stem cell therapies. And we don't want to hype anything. But in terms of how you feel about the field, I mean, there is a real clinical reality that is setting the stage for future developments, yes?
Absolutely. I think this is one of the exciting moments in the field, is how these basic discoveries have now led all the way to clinical translation. And I think the society is also very engaged in promoting guidelines that support this appropriately, public discussion, ethics perspectives, but also how, and I think at the meeting there are even some sessions really talking about how you go from basic science to translation. How do you talk with industry? How do you talk to regulatory groups? So this is as important as all of the other aspects. So it is a transformative moment in the field where hopefully we can eventually really help heal damaged tissues.
For basically a story about the 20-year birthday of induced pluripotent stem cells, I had the great privilege to chat with Shinya Yamanaka recently. And what was interesting when I asked him about that moment, that translation, and he didn't go into much detail, but I guess it's very hard to make stem cells ready for clinical application. Stem cells don't, one scientist has said to me once, they don't behave. Um, there are many ways that they differentiate. How do you deal with that in the lab? And I guess computationally, I don't know how you show that some stem cells are like, I'm done differentiating. I'm not going to go further. That's just who I am.
Yeah, so in my lab, we recently developed a new technology to deconvolute stem cell heterogeneity, and the field is aware of that different stem cell line behave differently, or even within the same stem cell line, specific clone behave differently. And recently, my lab developed a technology to isolate a specific clone, which has a specific like a fate propensity, and then start looking at the genomics, epigenomics inside those cells. And we started seeing sort of epigenetic characteristics in those clones. So reprogramming might not be perfect yet, and therefore creating heterogeneity and those clones behave differently. But having those like the view of heterogeneity, we may be able to in the future also regulate and control those heterogeneity.
And
So
those cells are basically genetically identical, right? In some cases, they're just,
yeah, we think they are genetically identical, but still have different propensities.
ouch. Okay.
I mean, I think that there are amazing technologies that are being developed now, but I think it's not just about profiling the cells. I think one has to test the functions of these cells to see how differentiated they are. So sometimes this involves using more complex in vitro models or in vivo models to really assess how differentiated a cell is. Have they adopted all of the mature characteristics?
Yeah. I mean, I'm sure that, this isn't your realm, but I do notice that there are stem cell clinics that offer and promise all kinds of things in the United States, in Europe, in Asia, in many countries. And if if you talk to people who have hopes about that, obviously they maybe have relatives who have had strokes, have neurodegenerative disease, or they're just really kind of interested in this kind of thing for various types of illnesses. What do you say to people who are like, well, that website looks kind of promising. Should we try it? Should we go there to country X and do this or not?
And I think a lot is about the providing patients or with information and accurate information. And so through the society, the ISSCR does have a website for patients to go to and understand the different terms, to understand what's viable, what's not viable, so they can make their own decisions. And I think a big effort of the society is also to try and advocate against these unproven stem cell therapies because they damage the field as well as people. So it's a really important topic, and just speaking with people, making sure that they're informed is a key part of this.
I saw that you have a patient speaking at the, let's see, our former patient. I guess she had an islet cell transplant, I think 20 years ago, if I'm understanding correctly. That's pretty cool. So you have patients also who are speaking at the conference and presenting, not just kind of present at the conference.
Absolutely, and I think this is one of the most inspiring aspects of the meeting is having either patients themselves or patient advocates talking in a plenary session. And so I think in our clinical session, we have not just the patient who is talking, but also we have people who are really at the forefront of clinical trials or of developing new strategies to try and address transplant issues and using genome editing to really solve difficult diseases. So putting these all together, I think will help people understand and be inspired to also really continue research.
Yeah, very cool. Let me just check to make sure there are no questions. Are there no? Am I seeing anybody? No. Um, do you think that the engineering aspects of sort of stem cell engineering, the different types of gene editing, is that an area that is very happening right now, or do you think it's kind of, steady, or have there been big spikes in activity? The types of submissions that you see, you probably have a good sense of what types of trends there are, let's say, engineering. In certain ways, it can be CRISPR or other types of gene editing.
Yeah, happening very well, and like engineering is important. We need to reverse engineer our object to understand what it is. And also, genome editing has lots of therapeutic potential. You can correct the mutation, and then use that stem cell reagent for therapy. I wouldn't create any hype on this, but that's the potential, and lots of researchers are working on this.
Right. And I think you have presentations on this, if I saw it correctly. Cool. So somebody who wants to, for example, dabble in this or is interested in this can find people to talk to. It's not like you're saying,
Absolutely, absolutely. So we will have fans on. I'm not sure if he's gonna talk about genome editing, but yeah, he does lots of interesting stuff.
yeah, unlikely that he won't talk about genome editing, right?
We also, I think, have lots of different talks on different aspects of engineering, biology, and new approaches in vitro to model some of these more complex in vivo niches. So I think this is another aspect of engineering that's really exciting at the moment in the field.
So given that, do you foresee or maybe want to predict any unexpected stem cell therapies or uses in the future? Just I don't know. I think no, just curious. Like, is there some one type of stem cell or one type of organ that's like Apart from I think the kind of usual ones, right? I think
Yeah.
pancreas is clear, for neurodegeneration is clear, but sort of anything particularly interesting or strange or that you would like to see, that you would like to see people move towards? I'm just curious.
So stem cell has the superpower of becoming, many different types of cells. But engineering stem cell could be a leapfrog because we have the potential of augment the function of stem cell and use them for diverse applications. So yeah, like a genome editing stem cell and augment the function could be a sort of crazy future.
Mm-hmm. Mm-hmm.
I mean, the idea, of course, of regeneration and rejuvenation is very compelling, right? If you could say, okay, this part of my body isn't working as great as I would like, or can it be rejuvenated? I mean, that is very promising and also problematic, probably. But I think it is fascinating too that there's the potential for that. Yes.
I think also that's one of the exciting areas of looking at non-model organisms who do have this amazing regenerative capacity. So what are the limits in humans or mammals, and how are these transcended in these other species, and what can we learn? And often these are very different pathways that are used, but maybe there are
So,
some
like
conserved principles.
limb regeneration or things like that.
Exactly, yeah. Yeah, so there are beautiful studies across many organisms from planaria to, axolotls and all different kinds of animals that we haven't even explored yet. So I think this can really bring surprises that actually can transform the field as well.
Yeah, they can do things that we can't, which is perhaps humbling and important for us to have that moment sometimes. It's kind of amazing how I guess, those surprises happen in labs. The other thing that I hear about from scientists is that working with stem cells, actually working with them in the lab, what is challenging is that they are so diverse and they don't actually, even though one can think that one has something in the dish, maybe they're not quite what you think they are in terms of state, cell state. Do you have recommendations when people are intrigued, like say PhD students who's starting out with stem cells and is frustrated by the fact that it's a Tuesday the experiment might go differently than Thursday?
I think the unexpected is always the best. So just embrace the unknown and where things science takes you. They are tricky. I think that but if we look at them and understand them, we can understand more about them. So I would just argue go where science takes you and think about it broadly.
Right. And they might build something that is an AI-based agent model of some kind, and their systems might actually offer some surprises too. I imagine there's kind of an interaction between wet lab and in silico lab there too, right?
Yeah, those struggles are always good. Um, you know, you face the struggle and try to be creative. I always like the story of Shinya Yamanaka discovered those like a transcription factor combination. Like they are testing one transcription factor one by one each, didn't work. And then two combinations, one by one didn't work. Three combination, now it's a combinatorial explosion. And Takahashi, like he put all the transcription factors, throw them in, one night, and then found the IPS. And then they started removing one transcription factor each to characterize which is unnecessary. And then, identify those like four transcription factors. So sometimes you start doing hard experiment and get inspired some crazy idea and it works. So
Yes, I asked them a little bit about that. I guess they were surprised, for instance, that nanog wasn't as influential as they thought it might end up being. So I guess you have to kind of keep an open mind about things because you're expecting certain things, but they actually don't come to pass. That's hard to teach. If you're a PhD student or your postdoc comes with their lab notebook, Shinya Yamanaka explained that when Kazu came to him with the notebook, then was skeptical, and he asked for the experiment to be done a few times, but then I think they started to believe. So I guess it's okay to be skeptical a little bit.
Very important to be skeptical as well. Yeah.
I have lots of skeptical students in my lab.
About their own results, like they're making findings, and then they have to.
Right.
I mean, truly, I think no PhD student doesn't have that moment where someone replicates their work and it's like, oh, good. Okay, it worked. It's true. What I found is true. I think everyone has that, like, yes, I did it. Someone else can do this. As that's just part of the process a little bit, is also becoming more confident in what you can provide, like what you can show.
Is there, I don't know internationally, are there good job outlooks for people in stem cell science? I mean, I assume that the clinical approvals in Japan are going to kind of send all sorts of effects across the field. And do you feel like that is important? And then I imagine also that basic researchers are like, awesome, clinical colleagues, we love you, but I'm actually personally interested in something very much more fundamental. So is there also maybe a tension that arises because there's a lot of clinical opportunity, but maybe not a lot of basic research? I'm just curious about the atmospheric things that you've seen and the submissions to you, but what you expect for discussions at the meeting.
I mean, I think one of the amazing things about the stem cell field is it does have this breadth and that you can go in many different directions. And so and move from basic science to translation if you want to, but you can also be engaged on different levels, think about things from a more ethics perspective, or become more involved in advocacy, in writing about science and doing science. So I think there's so many facets to stem cell biology that there is space for everybody. Of course, there's competition or tensions between things, but if by working together and appreciating these different dimensions, I think the field can really move forward much faster.
So job hunters are welcome at the meeting, but also that they're probably going to find out about some new things as well as sort of the established stem cell science, but also kind of these new areas, also interactions with AI or interactions with other types of emerging areas. And maybe it isn't a stem cell biology department that will ultimately hire them. Are you both in, what are the names of your departments again? I looked that up, but I was wondering. I don't think stem cell is in the,
No,
is,
I'm at the School of Biomedical Engineering.
And I'm in the Biocentrum or the Biocenter, where it's a very interdisciplinary group of scientists. Yeah.
I see. So that might be common too. Well, lovely.
Vivian, this is Shang from the ISSCR team. I'm the scientific programs manager. I want to put a plug here also on the special partnership we're doing this year with Canada's Stem Cell Network. It is a global workforce development. We will have a concurrent session at the meeting. It will be highly interactive, potentially some hands-on experience as well to gather input from the trainees on opportunities and challenges they are facing for career development.
Oh, I see. So
Yeah.
is that the one-on-one sessions that I think you also have where people can go, it's not
Oh,
the same thing.
it's not the same. This
I
is a
see.
special initiative of collaboration between Stem Cell Network, the Canadian Stem Cell Society, as well as ISSCR. They will actually have a meeting at the ISSCR annual meeting and then at the TMM meeting later in the year. The idea is really to gather input from global community on what are the challenges and opportunities for trainees for their career development.
Cool. And TMM stands for to.
That's the Canadian Stem Cell Society's annual meeting.
Right.
And so the one-on-one meetings where people can go, I think it sounds like it's also about spinning out and interacting with people and getting kind of input on pharma or collaboration with companies. Is that right? Fiona, you can tell me.
Yeah, there are all of these. So you have the one-on-one opportunities. You have these more hubs, if you science hubs where people, or scientific hubs where people can come together. They're actually, this year, one thing we really are excited about is we've created a large space with tables where people can really meet and talk to each other, because at some meetings it's been hard to find just physical space to meet. So it's kind of like a library where you're allowed to talk. You're encouraged to talk and meet. So really trying to provide actual physical places there.
Yes, of course, and the seating arrangements, or hopefully near a electrical outlet where you can charge your devices and all of that. They're often pretty rare to come by. I've learned that you have to sit on the floor near an electrical outlet. That's usually where conversations happen. Oh, and I see Tim is the Till and McCulloch meetings. I see. So that's the Canadian one. So that's one for people to keep an eye on. I can put a link to that. Let me just check again if anybody has any questions. We can keep you busy, but I know that you have other things to do than talk to editors with nothing to do. I'm speaking for myself here. I'm sure Angela has some things to do. Does anyone have any questions they want to ask? Any things they want to say? If not, we'll give our guests a closing moment. All set? All good? I have more questions, but we'll have that. Oh, yes. 2-year-old. Somebody's 2-year-old is running around. All good. Thank you. So, Fiona and Nozomu, do you have any closing wishes? Anything? Oh, right. I forgot to ask you about the manga. Yes. Your art on your website is wonderful. Do you draw?
Definitely go check out his website. It's very
Oh,
cool.
yeah, thank you very much. I hired somebody.
Oh,
That one. Yeah. She's a great artist. I don't know if you know anime called Spy Family, and then she was a part of the team in Spy Family movie. Um, I basically
Cool.
headhunted her on X. I
oh,
found her on X, and I started talking to her. Like, yeah. Anyways,
that's really cool.
thank
They're
you.
they're
It is very
very
cool, and it has a
very
kind
nice
of
website.
Yeah.
interesting vibe.
Yes, different than most scientific lab websites you find.
Thank you.
Definitely true.
So closing comment. I probably go brief, and then maybe Fiona can do more. As I said in the very beginning, stem cell is the sort of the hub of biology. I would say it generates many things, and I believe stem cell biology is the hub of biology too. And I hope that the stem cell generate diverse systems, and we can think in this way. This in the stem cell community, we have lots of people thinking about those like the generation of diverse systems, and outside of the stem cell field, there are other conferences with specialized topic, and we always want to link together. So I really hope that this conference, ISSCR, will be the hub of engaging many people from diverse area, and then think about what is the central system. Think about what is the central system. So
even if you're a math person or a computational person, but certainly
of
a math
course,
person,
yes.
and you are welcome. It's not because you don't
Yes,
know.
I'm pretty sure they will find very interesting problem in our conference.
And actually, we even this year, I forgot to mention this earlier, we have mathematical and theoretical speakers. So mathematical modeling and theoretical biology, because I think this is a really key part of understanding different forces in stem cell biology. So we're already trying to draw in the mathematicians to the meeting.
Theory allowed, math allowed, and of course, biology allowed. Well, lovely. Thank you so much for taking the time for this. And thank you, everyone who's been tuned in, even those, let's see, one colleague has a 2-year-old running around singing Frozen, which I probably think will have some copyright issues if we let the child sing. So we won't do that. But thank you very much for taking the time. Thank you, Dr. Doetsch and Dr. Yachie. Thank you, Angela, for co-moderating. Thank you, everyone, for tuning in.
Thank you,
Bye.
Vivian.
Yes, thank you so much. It was great to be here. We look forward to seeing you soon.
And that was Conversations with Scientists. Today's episode was a sneak peek of the upcoming meeting of the International Society for Stem Cell Research, ISSCR. Today's guests were the two program chairs of the meeting, Dr. Fiona Doetsch from the University of Basel and Dr. Nozomu Yachie from the University of British Columbia, who also has a lab at University of Osaka. Thank you to them, of course, also to Kim Kilborn, who helped set up this podcast. She is Director of Media and Strategic Communications at ISSCR. Dr. Xuang Du from ISSCR was in the podcast too. Today's co-host was Dr. Angela Parrish from Nature Communications. The music in this podcast is Rex Banner, Get This Party Started, Danny Shields, Now That We're Here, and Raw by Nonsense, all licensed from artlist.io. And I just wanted to say, because there's confusion about these things sometimes, nobody paid to be in this podcast, and the International Society for Stem Cell Research didn't pay for this podcast. This is independent journalism that I produce in my living room. You can find this podcast on streaming platforms and on YouTube in the channel Convos with Scientists. I'm Vivian Marks. Thanks for listening.